Hello students, I am teacher Svetlana and in our today's lecture we will learn about the mechanism of respiration. Part A, that is breathing, and B, external respiration, we have seen in our previous lecture. Today we will see Part C, internal respiration, and Part D, the cellular respiration, which is covered in the chapter Respiration and Circulation. So let's begin. Internal respiration. The two main components of blood involved in the transport of the respiratory gases, that is CO2 and O2, are your RBCs and the plasma. Transport of oxygen. Of the total oxygen transported, only 3% is transported in a dissolved state by the plasma. The remaining 97% is bound to hemoglobin Hb present in the RBCs. Hemoglobin acts as the respiratory carriers. It has high affinity for oxygen and combines with it to form oxyhemoglobin. Theoretically, one molecule of hemoglobin has 4 Fe+, each of which can pick up a molecule of oxygen, O2. So you have hemoglobin plus 4 oxygen molecules giving you oxyhemoglobin. Oxyhemoglobin is transported from the lungs to the tissue where it readily dissociates to release the oxygen. That is, your oxyhemoglobin Hb4O2 dissociates to give you Hb plus 4O2. So this is how most of the oxygen gets transported in the form of oxyhemoglobin. So basically the oxygen capacity of hemoglobin is it can carry 4 oxygen molecules at a time, and it can readily dissociate and release this oxygen molecule. However, the degree of saturation of hemoglobin with the oxygen depends upon the oxygen tension, which is the ppO2. 100% saturation is rare. Maximum saturation of 95 to 97% is at ppO2 in alveoli, that is at 100 mmHg. Degree of saturation decreases with the drop in ppO2. This begins the dissociation of HbO2. At 30 mmHg of ppO2, only 50% saturations can be maintained. The relationship between oxyhemoglobin saturation and oxygen tension, ppO2, is called as oxygen dissociation curve. This oxygen hemoglobin dissociation curve is a sigmoidal curve, and it shifts towards the right due to increase in H+ concentration. Increase in ppCO2 and rise in temperature and rise in DPG, that is 2,3-diphosphoglycerate, is formed in the RBCs during glycolysis. It lowers the affinity of hemoglobin for oxygen. Here students, this is the oxyhemoglobin dissociation curve, which is sigmoidal in shape. Now let us have a look at what is Bohr effect. It is a shift of oxyhemoglobin dissociation curve due to change in partial pressure of carbon dioxide in the blood. Halden's effect. Oxyhemoglobin functions as an acid. It decreases the pH of the blood. Due to increase in the number of H+ ions, HCO -3 changes into H2O and CO2. So you have in this Halden's effect, we see that due to increase in the number of H+ ions, we have HCO3, which changes into a molecule of water and carbon dioxide. In the alveoli, where the ppO2 is high and ppCO2 is low, oxygen binds with the hemoglobin, but in the tissues where the ppO2, that is your oxygen tension, is lower and the tension of carbon dioxide is high, the hemoglobin does not hold as much oxygen. It releases the oxygen for diffusion into the tissue cells. Carbon monoxide poisoning. Affinity of hemoglobin for carbon monoxide is about 250 times more than for oxygen. In the presence of carbon monoxide, hemoglobin readily combines to form a stable compound, carboxyhemoglobin. The hemoglobin is blocked by carbon monoxide, preventing oxygen from binding with hemoglobin. Thus, less hemoglobin is available for oxygen transport, depriving the cells of oxygen. This is called as carbon monoxide poisoning. Now students, let us have a look at transport of CO2. Carbon dioxide is readily soluble in water and is transported by the RBCs and the plasma membrane in three different forms. Firstly, by plasma in solution form, around 7%. Only 7% of the carbon dioxide is transported in the dissolved form as carbonic acid, which can break down into carbon dioxide and water. So here's the reaction: carbon dioxide plus water. It's a reversible reaction giving you carbonic acid. Then bicarbonate ions, 70%. Nearly 70% of the carbon dioxide released by the tissues diffuses in the plasma and then into the RBCs. In the RBCs, the CO2 combines with water in presence of a zinc-containing enzyme, carbonic anhydrase, to form carbonic acid. Carbonic anhydrase enzyme is found in the RBCs and not in the plasma. The rate of formation of carbonic acid inside the RBCs is very high as compared to its formation in the plasma. Now students, this carbonic acid being unstable almost immediately dissociates into HCO3 minus and H plus in the presence of the enzyme carbonic anhydrase, Ca, leading to large accumulations of HCO3 minus inside the RBCs. It thus moves out of the RBCs. This would bring about imbalance of the charge inside the RBCs. To maintain the ionic balance between the RBC and the plasma, chlorine diffuses into the RBCs. This movement of chloride ions is known as the chloride shift or the Hamburger's phenomenon. HCO3 minus that comes into the plasma joins the sodium potassium, forming NaHCO3 and KHCO3 to maintain the blood pressure. That is, they form the sodium bicarbonate and potassium bicarbonates. Here is the reaction: HCO3 minus plus Na plus gives you NaHCO3, that is sodium bicarbonate. H plus is taken up by the protein hemoglobin. So you have Hb plus H plus giving you HHb, that is a reduced hemoglobin. These H plus ions might be expected to lower the blood pH, but they are buffered by hemoglobin by the formation of deoxyhemoglobin, that is a reduced hemoglobin. At the level of the lungs, in response to the lower partial pressures of carbon dioxide, the ppCO2, that is the tension of carbon dioxide of the alveolar air, hydrogen ion and the bicarbonate ions recombine to form carbonic acid, and under the influence of carbonic anhydrase yields carbon dioxide and water. Now let us see the transport of carbon dioxide by the red blood cells, which is 23%. Carbon dioxide binds with the amino group of the hemoglobin and forms a loose bounded compound, that is carbamino hemoglobin. This molecule readily decomposes in regions where the partial pressure of carbon dioxide, that is the ppCO2, is lower in the alveolar region, releasing the carbon dioxide. So you have Hb plus CO2 giving you HbCO2, that is carbamino hemoglobin. Now students, let us have a look at the last category in the mechanism of respirations, that is cellular respiration. It is the last step taking place inside the cell where food is oxidized and ATP is generated. It can be shown by two major steps. The first is oxidation, that is breaking down of complex organic molecules into simple inorganic molecules with release of heat energy. That is, you have C6H12O6 plus 6O2 giving you 6 carbon dioxide molecules plus 6 water molecules plus an energy of 686 kilocalories. And the next important step is phosphorylation. It involves trapping the heat energy in the form of high energy bond of ATP molecules. So this heat energy that is generated, it is going to be trapped here into the high energy bonds of your adenosine triphosphate. ATP is used to carry out vital life and vital life activities, and so it is called as the energy currency of our cell. So the reaction goes ADP, that is adenosine diphosphate, plus an inorganic phosphate molecule IP plus 7.3 kilocalories of energy, giving you heat energy, giving you your ATP molecule. So this is called as cellular respiration, where energy is generated at the cellular levels. Thank you, students, for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe, and keep learning.